Muy bien. Bueno, muy buenas tardes a todos. Mi nombre es Soledad Ceruti y en nombre de la Sociedad Argentina de Espectrometría de Masa les doy la bienvenida a esta ya quinta charla del ciclo de 6 seminarios virtuales de la Sociedad. Tengo el agrado de ceder la palabra a la doctora Malena Mansi del Instituto Nacional de Tecnología Industrial, quien con su expertise en la temática estará a cargo de presentar y moderar la charla de esta tarde del doctor Timothy Garrett. Finalizada la presentación, tendremos espacio para las consultas que vayan dejando en YouTube y en Zoom. Cedo la palabra a vos, Malena. Muchas gracias.
Gracias, Soledad. Hello, good afternoon. It's an honor to present Dr. Timothy Garrett. As a short introduction, I would like to mention that after graduating in 2006, Dr. Garrett accepted a research assistant professorship at the University of Florida in the Department of Medicine, where he began research on targeted quantitation from clinical research and diagnostic using mass spectrometry. In 2014, he transitioned to an assistant professor in the Department of Pathology, Immunology, and Laboratory Medicine at the University of Florida, continuing his work to translate mass spectrometry to clinical areas. As a clinical member of pathology, he worked on the development of several new clinical assays utilizing mass spectrometry that are currently used in clinical toxicology. He's currently an associate professor in the Department of Pathology, where he's also the director of high-throughput metabolomics for the Southeast Center for Integrated Metabolomics, and conducts research in the application of metabolomics to disease diagnosis and clinical diagnostics. In addition, I would like to mention that the research in his group has led to over 100 peer-reviewed publications with more than 3,000 citations. Over those years, Dr. Garrett has developed technology, bioinformatics, and quality metrics in metabolomics, and today he's going to talk about advances in lipidomics and metabolomics for insight into disease mechanisms. Thank you very much, Dr. Garrett, for being here today, and it's all yours.
Wonderful. Thank you for that great introduction, and thank you for this opportunity to be here virtually with you all. And I'm excited about the opportunity to talk to you about some of this research and hopefully give you some insight into what we're working on and how we use mass spectrometry to try to bridge the gap between research and clinical medicine. So this talk is broken up to just a little bit of introduction to some of the stuff that we use, metabolomics and lipidomics. I know you may be aware of those, but just to sort of set the stage, to talk a little bit about some of the open source technology we've worked on, and then to give you some insight into 3 different areas that we've used this kind of work before. And so, as a first order of business. Yes, what is it?
Sorry, that is not in presentation mode. Maybe you can change it.
Is that better?
Yes, perfect. Sorry
right.
for the interruption.
No problem. Thank you for correcting it before I got too far on. All right. So we'll go and talk about the center. Part of my research involves working with the Southeast Center for Integrated Metabolomics. This was an NIH-funded center to improve our ability to do metabolomics and to also introduce metabolomics to a broader scientific community. We use both mass spec and NMR as a part of our center, which was a nice, unique incorporation. And one of our goals was to drive technology that would then improve how we do metabolomics in a really high throughput way. And I always like building tools that are open source so that other people can use them without having to purchase software tools. And in the past, we had grant opportunities for people to be able to do metabolomics. And we're at the end of that, but we hope to restart this pilot program in the future. And one of the key parts that I love is training people how to do metabolomics and being a part of their development, either as a graduate student or as a scientist, to learn new things. We've worked, this is just the United States, we've worked also around the world, but we of course are located in the state of Florida, right here where the star is, and we work with a lot of different people across the United States and several different countries in both training, but also in analyzing samples and producing results. So we are, of course, in the era of omics technology, and we've been in that era for a long time, with genomics really leading that way. and then proteomics following along with the advent of electrosporanization. So in this sort of cascade of omics, metabolomics is really considered sort of the last part of what happens, and it's what's happening immediately. And so we think of this as the omic that's most responsible for the phenotype that you see right away. So it has the opportunity to measure as well as represent what's happening to everybody on a daily basis. And we use several different terms in this field. Metabolome really is the collection of small molecules that might be present in a cell or in a given system. And while metabolomics is the measure of those metabolites, right? Using technology to measure what's in there. And what I'm really going to talk about is really the ability to do potentially comprehensive characterization of either lipids or small molecules. Although we know we can't measure everything, we try to get as many as we can. Your metabolites and your lipids are a result of your genes. They are a result of how old you are, and you do have differences between your chronological age and biological age, depending on what might happen in either your environment or other stressors. You also, if you are sick, whether that's an acute illness or whether that is a chronic illness, those changes are reflected in your metabolites. And of course, if you take either over-the-counter drugs or nutraceuticals or anything like that, those will appear in your metabolism. And the food you eat and the exercise or other things, sleep that you have, all sort of reflect this, right? And of course, the time of day that you sample the metabolome can also have a difference in its measurement, which is why it represents us on a given daily basis. And that diversity is important, but we also can use this diversity to help improve our biological variation when we measure diseases. And so I'll give you an example of how that diversity can be really helpful in trying to identify biomarkers. And a subset of the metabolome is the lipidome, right? And so the lipids are involved in many different diseases. They've been implicated in virus replication, and we did a study on this to understand COVID transmission related to how it utilizes lipids, but they've also been involved in other kinds of viral infections. Most of us think of lipids as the use of energy or storage, where we utilize what's left there to build other things, either in exercise or other areas. But they're also involved in signaling, and they form the cell membranes, and of course, many other areas. One of the downsides is that they are ubiquitous, meaning that they're very common across species, but they're also some lipids that are very unique, like, for example, lipid A that might be in bacteria. And of course, measuring these, just like the metabolome, is very important to understand potential biological processes. The cool thing is that lipids actually consume a huge portion by weight of plasma, right? So we focus a lot on these kind of small molecules, from sugars to amino acids, but the lipids can cover a majority of what we might see in a plasma sample. So they're very important in terms of what that constituents are. And then, of course, they're very diverse across multiple species, whether those are sterile lipids like cholesterol or glycerophospholipids like phosphatidylcholine. And of course, even in a given class, they have a high degree of variability, right? And so this is just a picture of what kinds of lipids might be present, with those based on the glycerol backbone, or those that are based on the sterile backbone, like, as I said, cholesterol. Then you can also have alcohol connections or ester bonds. And of course, with phospholipids, you have a head group and a tail, the two fatty acid tails, and typically a hydrophilic head group. While in triglycerides, you have a glycerol backbone and three tails, or a diglyceride with the removal of one of those fatty acid chains. And looking in just a given class, say we look in the glycerophospholipid for triglycerides and mono and di, as we convert in between these, we can either build up to add a fatty acyl chain, or we can take them down. If we look just at a triglyceride and we vary the number of carbons from, say, 20 to 24 carbons with degrees of unsaturation, we can get a large number of potential lipids that could be present, over 10,000 possibilities within just a single lipid class. And this is part of that diversity that's present, and also part of the difficulty in measuring is we can't measure all of those 10,000 variability possibilities in a given system yet, but we can get a lot of information that at least allows us to get good, clean information related to what kinds of lipids are there. And so the cool part about lipids is that we have the ability to identify them really well using tandem mass spectrometry. And so we have fragmentation patterns within MSMS that allows us to differentiate all the classes of lipids. So this allows us to filter MSMS data or to create software tools that can be a key part in differentiating what's there, as well as being able to clearly identify what tails are present. And so there are a number of different tools, many that are free and some that are paid for, that do this sort of process where you use mass to charge and MSMS patterns to help identify what's there. This first one is the one we developed as an open source tool that runs in R. And then you have these other different aspects, as well as the different capabilities of each one. The tool that we developed, of course, interfaces with multiple different open source platforms to compile the data. So you still have to compile the data across multiple samples and then run the data through Lipid to do the identification process. And it works with any vendor format as well, from what we've tested Agilent, SciEx Waters, and Thermo systems to run through this system. So it works really well with both all ion fragmentation as well as MSMS. And of course, the libraries are open source, so you can add libraries or take away libraries given what's going on. And so right now, at least roughly 300,000 in silico species present across 60 different lipid classes, and grows, of course, as you want to. And it can run on any platform, which is really good, at least in our case, trying to keep it as open source as possible. A simple workflow of this is that we have an acquisition. We then use a program to pick peaks like MZMine. And then we use the MSMS data from this acquisition to identify lipids and match them back across all the samples. And then those annotations get appended, and then we can then do our statistical analyses, right? You can also do this with imaging or direct infusion kinds of experiments if you want. Of course, when you do that, you lose chromatography, but that's okay. You gain speed in that kind of process. A little bit of the detail that goes into it, just as you can see, we have potential isomers that could be present, and they can be separated by chromatography. And so, of course, we could have to keep the retention time that goes with that. And then, of course, keep the fragmentation patterns under each window. And so that's what we get in this case. We then get a table of different retention times that match different fragmentation patterns. And then we work through the intensities, and then eventually get down to a process where we identify them based on how we can identify them, either by a very robust way, with being one where we can identify the fatty acid chains and the class, or we have the same process where 2 and 1 are really the same, but we're using a data-independent analysis instead of a data-dependent analysis. And then, of course, we can identify only the lipid class if we don't have all the fragmentation pattern, and we label that as a 3. And then those that are unknown, don't have any match for MSMS, or we didn't get MSMS on, we would label those as a 4, because at least they match something in a database that could be a lipid, and we want to at least keep it present. Yeah, and across, a cool part about it too is that you can also use the intensity of the fragmentation information to rank them based on order of intensity across a given chromatogram. Because we have isomers that are present in lipid world, we have to sort them in some way, and so we decided to sort based on intensity, and that at least gives us some sort of rank to know which one is the most abundant, and then we can go from there, right? The key part is, of course, getting a fragmentation information at the most intense part of all peaks. And of course, you could have some errors with that because there of any slight shift in retention time that can happen. And since we're relying on MSMS, we have to be able to gather enough tandem mass spectrometry without reducing our ability to capture full scan data as well, right? And so in a typical process, we use something called data-dependent analysis, where at a given peak, that's eluting, we get a mass spectrum, and we would use, say, a data-dependent top 5 experiment where we might take the top 5 of these peaks, and those would then perform MSMS on, right? But we would miss all these other peaks, and if we repeated the same process, we would always get the same most abundant lipids, and we'd always miss the less abundant lipid species across the entire chromatogram. And so if you can iterate this process and take this injection and then eliminate these peaks for the next MSMS experiment, and then you can finally get down to this next level of lipids that are still important but aren't as intense as the most abundant species. And we call this iterative exclusion, or IEomics. where we can do this up to whatever number of times. We typically see you need to do it anywhere between 4 and 8 times to get a good estimate of what's going on within the system. And that, of course, depends on the concentration of the sample. And you can sort of see this in how it creates a better picture of the lipidome, where we have the MSMS collected across a entire experiment where we would get lots of fragmentation information. But if we do this in an iterative way, where each run has a new type of MSMS that's run with it, we can then get a much more improved density plot of what's really present in a given sample, right? And this translates, of course, better confident IDs, where we get more injections per sample. But really, it translates into less bias, right? So if we look on the left side, we have plasma that's done just in normal data-dependent mode, and then we add iterative exclusion. This is the number of unique experiments we get. So we get a lot more oxidized triglycerides in one, and a lot more diglycerides, right, which are less abundant species. So that proves to us that we're getting a better representation of the lipids that are present in this sample. And the same can be true for tissue samples such as substantia nigra. And in this case, the new IDs we're getting represent more phosphatoethanolamine, because we captured most of the PCs in the first types of experiments. And so this would then allow us to really reduce any kind of bias towards biomarker identification, because now we're able to represent what we can really ID in a proper way. Okay, so with that as a sort of direction, I'm going to switch to talking about some biological examples. The first one I'll talk about is really urinary cancer metabolomics, but it's a focus on prostate cancer, but then building in how we can use this metabolomics to really do good biomarker discovery. And so if you aren't aware, these are statistics a little bit old from 2018, where prostate cancer is the second most common cancer among men in the United States. And it is, of course, one of the leading causes of death in males in the United States. And of course, it occurs by the production of cancer that forms in the prostate of males. And so the first question we have is, one, that there are good potential treatment rates for prostate cancer, right? There is a high success rate to eliminate cancer in patients, but there's also a high overtreatment rate. We rely on an assay called PSA, which stands for prostate-specific antigen, which is, of course, a protein that's typically present in the prostate, but it's not really as reliable as we thought it would be. Screening doesn't correlate to stage of cancer, which is one of the problems, but also you could have a high level of PSA and still not have cancer, because that level doesn't necessarily correlate directly to any kind of cancer. And so really PSA has to be measured on a regular basis to show when you elevate, not necessarily on a one-time analysis. That's one of the issues that happens with it. And overtreatment means that you do unnecessary biopsies, which can have other side effects. Anytime you biopsy tissue, it causes damage, and it's and then causes the patient to also have other kinds of psychological fear about what might be present for them. And so the goal of this was to whether or not we could go back and use a non-invasive kind of sample to detect prostate cancer, and whether we could use urine as that medium, right? And so urine is successful. I always like to show this because urine is the oldest diagnostic really we have. Using this Pinder wheel from the 1500s that was used to diagnose, or at least not diagnose, but to figure out what might be happening in different individuals, right? So these are different colors of urine that would be indicative of, say, either metal poisoning or blood in your urine, or and so they would use visual cues. They would also smell their urine as a way to say what might be present based on the odor. And they would also taste it to see if it had any kind of different flavor to it that might tell you something unusual. And so sugar, in one case, might tell you that a person could be diabetic, even though at that time they didn't know that. The good thing is we don't have to use our senses, taste, smell, or sight to diagnose disorders. We can use high-level analytical chemistry. In this case, I'm showing NMR as a way to do that, but I'm going to talk really the rest about mass spec and how we use this in this area. All of our, as a person who's worked in the clinical lab, I like to make all of our methods as open as possible. So actually, almost all of our methods are uploaded on a place called metabolomicsworkbench.org, mainly because I just want people to see if we're doing something wrong, they can tell me and we can fix it. But this is just a simple example, simple workflow of taking urine, filtering it, and then adding a little bit of bacteriostat and a little organic material to help get rid of any potential proteins, and then drying it down and then bringing it back up to a volume that we could then use for on an instrument such as an Orbitrap. And this is the instrument that we use for this. And this is reverse phase separation, and our gradient is listed there. And of course, you can get that from me anytime you want it. And if we look at a simple test, right, we can take sample A versus sample B, right? And we have controls. Any good analytical chemist will include multiple kinds of controls. So we have a separate kind of quality control, which is just the amino acids, versus a separate quality that might measure our instrument performance. And then we could say I have a group of healthy patients and a group that has prostate cancer, right? And so it's that simple, right? We have, using principal components analysis, we can really identify patients that are different, right? So we can see prostate cancer versus healthy, clearly distinguished, and not a huge group, but we have a fairly good separation between them and fairly reproducible. And our quality control was good, right? But we have this one paper that came out in 2012, which basically says that only 1% of biomarker assays published in research translate to clinical diagnostics. And the translation is from a research grade assay all the way to a clinical test that the insurance company would pay for to diagnose a patient, right? And so what happens, one of my reasons is that we don't include the right kinds of specimens that would come to a clinical lab in our initial work to give us enough confidence to know that we are really doing the right thing, right? So it's cancer versus control appears to be very easy because that's what most of the papers are published on, right? But if we want a disease diagnostic test that allows us to separate multiple kinds of diseases and then really pick out which cancer it is, then we would need to have more samples that are representative of what we might capture, right? So we can move out to not just prostate cancer, right? And we can look at other kinds of disorders that might happen in a prostate tissue, right? And there's two main kinds of disorders that might happen, and those are prostatitis or benign hyperplasia, benign prostatic hyperplasia, or easier to say BPH. And those are both inflammatory events that happen and are very common of our patient who might come into the clinic experiencing issues that could be consistent with prostate cancer, right? And so you want to be able to have samples that are representative of those specific disorders, so you have a better way to differentiate patients that are really truly have prostate cancer. And so if we look at that and we run metabolomics, running an untargeted analysis, and we then compile everything, and of course we run everything randomized and blinded, and then we unblind ourselves later, we can then see that we can clearly separate those that are normal from those that have prostate cancer still, right? So that's good. And then we can easily separate those that have prostatitis and BPH down the middle, right? We have one that may fall into the window of prostate cancer, right? And then maybe one that falls normal. And we can't separate prostatitis from BPH, but at least we can differentiate an inflammatory event from prostate cancer. And prostate cancer can cause inflammation as well. So we have a double-edged sword here with what happened in the tissue. And we can look at individual metabolites, which is, of course, what we want to do in metabolomics, right? And so in our given assay, we cannot separate beta-alanine and sarcosine. They are isomers, as well as alanine. And so if we look at an expression, we can see that this would not be a great marker in urine for a separation of cancer versus BPH or prostatitis, right? It might separate normal, but it's not good for a disease diagnostic. Creatine, another common metabolite that's measured in urine. And I will note that we don't normalize to create creatine or creatinine in our metabolomics of urine. We normalize to either specific gravity or we normalize to the total useful signal because creatinine can change in patients. And this is one reason, right? In the normal patient, it's low, and in patients that have cancer, it's high, right? And if we normalize that out, we would normalize this effect away. But we also see that it's also high in BPH and prostatitis. While rather we look at glutamine, glutamine does look like it's much more distinguishing factor between those patients that have cancer versus those that are normal or those that have inflammatory events. And there is a significant amount of literature that has rested on using that tumors use glutamine for growth, right? And so you could potentially make a case that the depletion in urine is because the cancer cells require that to help them grow. There was a paper that published several years ago about the sarcosine, and you can see sarcosine is very distinguishing in cancer, but it also failed to translate all the way to a clinical diagnostic test, right? And so that this could be one of the failures where you have a lot of missed samples that would cause a misdiagnosis. And so this gives us a better picture of prostate health, right? And we also have things like you can look at normal from data when you have lots of samples you're running. So you can find samples that are signals that are related to normal versus abnormal, right? And that would be cool to just be able to distinguish normality from abnormality. And so that's it, right? We can stop at this stage and think about metabolomics and say, now we can translate this test, right? And then go from say hundreds of samples to thousands of samples. And then we're gonna translate this to a really thing that makes sense, right? Before we do that, it's probably better though, and I'm sort of an advocate for this than I am at going much larger numbers, is to increase our diversity of the kinds of samples that we might see before we really start to scale up to running thousands and thousands of samples, because that cost goes up exponentially when we get to that level. And so these diagnostic metabolites that we have could be shared across many multiple inputs and outputs related to metabolic processes. And that's one of the things with metabolomics is that, yes, we turn on the enzymes, we turn them off, and then we have to be much more specific if we can be, so that we can create these better diagnostic patterns, right? So if we can think about other potential types of samples we might need to build a better cohort so that we have this pilot study that then becomes much stronger, we would want to include samples that might come from patients who have bladder or kidney cancer, because those patients would definitely come in with other issues. We don't have a huge issue with diagnosing necessarily patients with bladder or kidney cancer, but because we get those samples in, we need to be sure that we can distinguish those subjects from others. And if we look at that, we can look at a heat map of these metabolites with a few identified. We have prostate cancer clearly clustering away from all the other kinds of samples, whether those be normal, bladder, or kidney cancer. And we can go back to our marker for creatine, and we can see that its signal is strong. It's still high in prostate cancer, which was the same as before. And then we can expand it to looking at BPH and prostatitis, and we still see that it's elevated, but it's not elevated in other cancers, right? So that might make it a unique marker for prostatic disorder, or at least metabolites that are unique to something going wrong with the prostate. And you can look at a different way to look at this. This is principal components analysis, so an unsupervised clustering approach that then allows us to really differentiate the types of samples we have here. And in the normal, we have both males and females in this, which is why we have a little bit of clustering differences between them. And if we just wanted to use this as a way to say, can we differentiate and remember, urine goes through the prostate, but also through the bladder and the kidneys, right? And so it rests through all of these different matrices, right? And so ideally, if we collect urine, it looks as though using this type of samples, we can clearly say, yes, we can know if you have something wrong with your prostate versus if you don't, right? And that would be, say, a first order test where we could collect urine, screen, and really confirm whether or not, yes, you have a prostatic issue that we would need to address, right? And then subsequently, we can then take that data and be much more confident now that we can separate prostate cancer from other inflammatory disorders. So this would be an example of, say, a second tier test using the same exact sample that was collected. And now we can find two unknown markers that are very unique to prostate cancer, one low and one high. And I keep these as unknown because we don't know what they are yet. And we're working to identify them, but they are very unique and potentially could represent a ratiometric way to identify prostate cancer. And of course, they have a really strong prediction rate if we just take the first top 10, including these two, and we can get a very high classification and prediction rate of this. We also can find markers once we add in risk, and this is using Gleason Score. If you're not familiar with that, Gleason scoring is a way to assess how dangerous or how metastatic the tissue is. And so 9 would be the worst, and 6, of course, would be more benign. And so we can find, and this is a marker of thymidine glycol that appears to be at least somewhat calibrative to help identify risk. And that at least allows us to have, say, not just the same. So again, this is the same sample that could be measured. So we don't have to collect a new sample. It just goes through a series of 3 different tests using mass spectrometry to then help really assign whether you have a prostatic disorder, what type of disorder you have, and then what risk factor you have. And then I'll talk a little bit about the unknowns in metabolomics just in 2 slides. Unknowns are very important. I showed you 2 unknowns that we don't know what they are yet, but it's important for us to spend time trying to figure out what they are. Because in the past, we thought that we had figured out metabolism, right? In the 60s, we thought it was done, and then we did move on to other things. And that's when, and then after 20 years later or so, proteomics blossomed, and we spent so much time on proteomics. But we really have to think about how we identify metabolites right, and how we expand our view of what types of metabolites we can detect, so that we get a much better view of the samples that we have, and also the unique metabolites that might be coming from different systems. And so this is just tackling this dark matter. It requires a community effort, and it requires you sharing data, right, that comes from experiments, so that people can use your data to help in the process. So I'll summarize this part of the talk and then move on to the other 2 disorders in the next section. That really says, this is a summary of sort of what we can do. Metabolites are excreted from multiple systems in the body to eliminate metabolites we don't want. And we can use metabolites from the urine to differentiate different types of disorders, those that might arise from the kidney or the bladder versus those that arise from the prostate. And then we can also assign risk. And so we're in the middle of developing a larger scale study now that we have this cohort. That the only other one we didn't have in here was patients with bacterial infections, which we will include in the next cohort. So then another project that we have that's directly connected to the clinic is a study on meningioma. Meningioma is a non-cancerous, typically a non-cancerous tumor that arises in the brain tissue surrounding both the brain and the spinal cord. And here's an example of that kind of brain tumor. You might be more familiar with glioblastoma, which is the much more severe form of brain cancer. But meningiomas are still very common. About 37% of brain tumors in the United States are meningioma grade. And they're classified in 3 different classifications. Grade 1 would be not very severe, and this is the highest grade, right? And so it's more benign versus those 2 and 3, which do have much more concern and of course require radiotherapy, not just removal. So chemotherapy associated with that, right? And so our goal would be to improve. One of the issues is how they differentiate grade 1 and 2 and 3, right? We still have some issue with grading one versus the other. And their specificity from tissue pathology is not as strong as it could be, right? So that's one of our disorders. The second one might be ability to differentiate grade 2 versus grade 3, but that requires, of course, a significant number of samples, and we only have, say, a 1 to 3% frequency rate of grade 3, so it's harder to get the number of samples you might need for this one. And so what we did is we actually classified 2 and 3 as one group, as higher grade versus lower grade. So we had enough to be able to differentiate those and to be able to at least get some insight into what kind of biomarkers they are. That might be able to do this kind of differentiation, right? And then, of course, ideally, we'd like to be able to identify therapeutic targets. So with lipids or metabolites that might be indicative of how we could treat meningioma. But in the first stage is really just doing a much better idea of what's there. And this, of course, is a connection to anatomical pathology here at the University of Florida with Jesse Kresak leading that effort. And sort of an overview of that, we had roughly 40 grade 1 versus 45 grade 2 and grade 3 tumors. These are, of course, extracted by 2 methods in this case. We did both a metabolite and lipid extraction, and then we ran those separately on the same instrument platform, then a lot of statistical analysis. And then, of course, we used artificial intelligence to build a better way to potentially identify markers from large feature sets. And I'll sort of tell you about both of those different instances, right? And so this is the breakdown of grade 1, 2, and 3. And again, we combine 2 and 3 together. And of course, the details about lipidomics, metabolomics, and you're welcome to have those details anytime. And of course, we always measure quality control and make sure that we have less than 15% RSD across all our runs before we do anything. And the reason for that is because for every day you spend on the instrument, you spend at least 7 days working with the data, right? So you want to make sure your data on the instrument is robust, so you're not wasting time doing statistical analysis on non-reproducible mass spectrum data. Again, just a picture of the instrument. Two things to note: we used MZ-Mine for both lipids and metabolites, but we use Lipid Match for identification, and we use an in-house library of metabolites to identify our first order metabolites with a high level of confidence, and then we do some other things to identify other ones. We, of course, once you now combine both lipids and metabolites, you have a very large data set representing multiple kinds of things going on. And the data is very nonlinear in nature. And so we have to think about ways in which we could potentially couple other ways to do biomarker analysis that allows us to at least deal with this kind of large environment. And if you look at machine learning and metabolomics, it's really blossomed, and there's a huge initiative in the United States driven by the NIH and other institutes to grow artificial intelligence and other machine learning and other approaches into biomedical science. And that's sort of why you see this explosion. And I would expect this to double or triple in the next year. So if we just look at the lipidomic expression from meningioma samples, right? So is heat map produced just at lipids? Again, we're separating just grade 1 in green versus grade 2 and 3 in red. And we can get one major cluster of triglycerides that seem to cluster really well and associate with a high level of expression in grade 2 and grade 3 samples and lower in grade 1. And if we pull out and look at this dot plot, we can get an assessment of the types of lipids that are present. And so what we observe in this one is that we do see a higher level of triglycerides in grade 2 and grade 3 that are related to longer chain but less degrees of unsaturation. So less double bonds, but so basically lower on this end but higher on this end in this dot plot. So this is where our intensity would lie. And so we think this is related to direct cancer growth, which is the ability of cancers or the need of cancers to get fatty acids from the lipogenesis process in the tissue and to assimilate those into the tumor to continue to provide resources for the tumor to grow. And that could be one of the reasons why those triglycerides, especially the unsaturation, is there. And we can then look at a bigger picture of all the lipid classes too. We can create these donut plots that at least gives us some sort of distribution between 2 and 3 and grade 1. You can see a change in sphingomyelin between them. And we can also then pull out individual classes, and we can see the change in acylcarnitines is unique, where it's elevated in grade 2 and grade 3 samples. We can also look at a bigger plot of all the lipids, at least showing the log fold change versus p-value. And I know this one here, hexosylceramide is not just, and this is a tissue sample, right? And so we're taking all these lipids across a different tissue sample. Hexosylceramides, which are glucosyl-based or sugar-based lipids, have an expression where they have a high fold change in grade 2 and grade 3. And if excuse me, a little bit ahead of myself in a second. I'll talk to you about this one in the next slide as well, as it correlates with another disorder. We also have phosphatidylethanolamine and phosphatidylcholine lipids as, of course, the biggest types of lipids present in this example. And we can look at the PC to PE ratio, which gives us some sort of idea of the fluidity of a membrane, right? But this last one, this hexosylceramide, actually correlates well with a separate study, a separate brain cancer type that's present in children, primarily called medulloblastoma. And this is CSF, not brain tissue, right? And so what we observed was an increase in hexosylceramides in the brain tissue. And in this case, we see a decrease of hexosylceramides in the CSF, right? And so correlated with the same tumor. So it could be that this hexosylceramide is trafficked out of the CSF and contributes to this tumor growth in the tissue organ, and this is where it's getting its resource. So that could be one way in this to prevent cancer growth. That could be potential targets for that. And this was, of course, a separate study where we really wanted to look at what would happen in CSF, so we could translate that, right? We can go back to the meningioma study, we could look at the metabolites, and we can also get an idea of what's going on with these metabolites. And the key part here is we're looking, of course, at a lot of purine biosynthesis that's happening, and we could sort of see how purines are decreased across the subset of grade 1 tumors, and they connect back to a series of other purine metabolites. And subsequently, we also found a connection between vitamin B metabolism, which was quite interesting in that it's downregulated in grade 2 and grade 3. We don't yet know why, but this is consistent with other papers where we see this pyridoxal and other vitamin B metabolites related to tumor growth. And so at this point, we really wanted to use other tools to, we could use that, we could dig through the data, and we could do a biochemical characterization of what's going on related to the metabolites and the lipids. But we also sort of really wanted, the goal of this was to develop a biomarker, right? And so the student, of course, who's been doing all this work was Hoda. She then harnessed the use of machine learning as a way to sort of use this and potentially build a better way to find biomarkers, right? And so we're using this basic process to test data, to loop it, and continually going on to be able to sort of apply it to this case, right? And so when she did this, we had roughly 17,000 features across metabolites and lipids. And so this was just classified as either lipid from positive ion with the mass and the retention time, or a metabolite and vice versa. So you can sort of see how we classify that. What we first took at was trying to figure out how to filter the data to get to a point where we have a reasonable number of metabolites or lipids to look at. You can't really do machine learning on 17,000 features, so we have to filter it. And in this case, Hoda took several different machine learning algorithms, and using these top 30 feature ranks based on this feature ranking system, where once you get lower, they have less significance, right? So we can see there's a pretty good cutoff, and we could choose a little lower, but this was a good inflection point to pick out what we might pick for doing additional machine learning, right? And so we took this data, threw it through several different machine learning algorithms, and really settled on the support vector machine learning as the best approach, which approximated roughly 3 different features at this level. And of course, this is the number of features. So the more features you got, the more it leveled off, right? But this N of 3 would allow us at least to spend some time doing identification on a smaller subset of species and a larger subset. And identification takes a significant amount of time. And so we took those 3 lipids, and we, of course, didn't have any of them identified, and that's okay. And so we used the mass spectrometry tools to help identify parts of this, right? First, we wanted to assess that, yes, indeed, our machine learning algorithm did pick samples that were predictive. And so you see these 3 features, they're all increased in grade 3 and grade 2, which gave us confidence to then say, yeah, let's spend some time identifying them, right? And so this is where we are in metabolomics, where we often get exact mass hits that don't mean anything to us until we can get other information about that. We could potentially get a molecular formula from the exact mass, and that could give us at least some sort of idea of what kinds of structures could be possible. We could collect MS on those to try to at least build pieces together, and then we could add a putative structure based on how those are built together. And then, of course, hopefully get down to this validated part, right? And so what these are, I'll tell you an example of how we did this in one of them. But essentially, these are the 3 identifications that we made. The first ended up being a triglyceride. And you would think that since we had metabolomics and lipidomics and we ran iomics, we ran a bunch of stuff, we'd identify this. But it turns out this is a very unusual adduct of a specific triglyceride. And so because it has a different mass spectral adduct, it wasn't identified properly. And that's how we got to this point, right? And then, of course, this galactosylamide, which is part of those hexosaceramides, which were elevated in the tissue, as I talked about earlier, also came out to be identified here. And again, this is a lipid, we should be able to identify it, but it actually had another strange adduct that forms to it. And the final one is a peptide, a metabolite, really, but it's a peptidomic metabolite that actually consisted of 14 amino acids, and this was identified through de novo sequencing. We don't have a parent of that peptide in terms of a parent protein yet, and it doesn't match anything in a database. So it could either be from a novel protein or it could be from a novel peptide production. But an example is 830. Of course, we want to validate that this is a real isotopic structure, and so we use the mass spectral pattern to do that, to see that it does have an isotopic pattern, it does have a true chromatographic performance, which is good. And then this is where the MSMS is very important. But what happened here is this adduct is a formic acid adduct that we get, right? And so MSMS is not as good for doing de novo sequencing on an adduct like formic acid. And so because what happens is you lose the formic acid, you get M-H. And then this is really what one of the diagnostic ions, you get that, but you lose some of the fatty acyl chain assignments, right? So we did pseudo MS3 by increasing the source collision energy to help identify these exact tails, and that's how we get down to this galactosyl ceramide with this fatty acyl region. And it's a high degree of unsaturation. Again, the biology of this we don't yet know, but this at least gives us a confidence that we have markers that we can identify, and now we can tackle how well they are. And so that work I showed you combined machine learning with metabolites and lipids to identify several unknowns. I showed you 3 of the 5 that we have. 2 of them were lipids, 1 was a small metabolite peptide, and we can't differentiate 2 from 3 due to the small sample sizes. But as we get more samples, we might be able to do that. And of course, our biological interpretation is ongoing with this part of it. We think we understand what's happening with the triglycerides as the tumors are using those for growth, but the galactosyl ceramides, we don't know for sure. So this last sort of 4 minutes, I'm going to give you a N of 1 study where a single sample from a clinical pathology was provided. This is from a 57-year-old female who had protein expressed in their urine and had a high history of diabetes as well as breast cancer, and had several different symptoms related to potentially what could be part of chemotherapy, but we don't know for sure for that one, right? They did think that the protein that was secondary to diabetes, as it tended to be more severe than what happened in her history past. There was no family history of any renal disorder or genetic disease, not generic, a genetic disease. And so because she was having issues with protein in her urine, a renal biopsy was performed to see if there was anything going on with her kidneys. One of the things that could be happening is this patient could have Fabry disease. Fabry disease is a disorder of missing alpha galactosidase A. And what happens with females is that over 60% of females are missed because they have some residual levels of alpha galactosidase A, and it can have late onset in life. Typically, diagnosis is made by using lyso-Gb3, which is a glucosolipid. And when this was run, the patient fell into this category. So it had a potential to have Fabry disease, but you would have to perform genetic testing, right? And at this point, when you have this suspected clinical disorder, you have a very huge rubric to go through to try to diagnose this patient, whether you get if you could do gene mutation analysis, which takes time, or you could do other things. So at this point, since the colleague is here at the University of Florida, he wanted to ask me if we could measure additional lipids, maybe to might give us some insight into what's going on with this individual, because of there are over 1,000 known mutations of this GLA gene, which causes changes in alpha galactosidase A. And metabolomics and lipids has been employed in many rare disorders now, improving our ability to dissect a greater part of the metabolome. So this is sort of part of a good area where we think we can have some strong impact directly clinically. If we look at the pathway of what happens in Fabry disease, these are the lipids that are involved, and this is the enzyme alpha galactosidase A. It actually impacts 3 different and separate processes of lipid breakdown. You have this global triosyl ceramide, GB3, where it breaks it down into 2 sugar, it takes off this alpha sugar. Again, you have a lyso-Gb3, which is the loss of a fatty acyl chain. And then again, this enzyme does the same conversion. But you have a secondary pathway that could be a different gene, or a different type of this enzyme that breaks down GA2, which is basically a 2-sugar head group on the tail, but it still has this alpha linkage to make it back into ceramide. And they all produce this ceramide. But if I'm a mass spectrometrist, I look at this pathway and I say all of these are capable of being measured. And so why aren't we measuring all of these to sort of see how the different enzymes are affecting the different pathways? And so that's indeed what we did. We still measured these, but we also measured and expanded to GA2. And I'll note that when we say 1, 2, and 3, they're more than just 1, 2, and 3, right? Because we have different fatty acyl chains that change, right? And you'll see that how many, as we show an example of that. So this sample is the exact sample that was given to us. We have the tiny sample in the middle, which is roughly 2 milligrams of tissue. We cut it out of the OCT medium and then extracted it only for lipids, because that's all we cared about. And then this would be a measurement of GB3, where we in the patient, we don't detect it, and that's what you would use if it's not detected. That means the patient doesn't have Fabry disease. Also, and we ran 2 controls. We also looked at that lyso-GB3, and we see the same result where we have nothing detected, right? So that proves, at least in this case, that the patient doesn't have Fabry disease. But the patient, of course, still has issues, right? And so as we expand to measuring GA2, we can see that we now have a marker that's much more distinct that could be related to a different genetic mutation that this patient has, because now we see an elevation of GA2 across 8 different GA2 species. And we have all sort of controls to go with that. And in almost all cases, actually in every case, the patient is elevated, but we have in some cases where the normal patients have an elevation just as a normal process. And so these results would confirm that this patient does have a variant of Fabry disease that seems to only be much more affecting of the GA2. And the accumulation of these is what really causes problems in the kidneys. And so, so measuring this would help us understand this different genetic variant, right? And we can confirm, yes, indeed, the patient has an elevation by looking at controls, looking at the mass spectra, and also doing MSMS. And then it gets us to this point of how you diagnose an N of 1, right? This is one patient. We have to be confident that that patient is indeed has this change. So we use something that we could use is a ratiometric process, right? So we might want to measure the ratio of the precursor to the enzymatic product of that, right? And so we could measure GA2, which to the ceramide, which is actually two processes down. So ceramide would be the base, and we measured this GA2 to the base metabolism. And this might give us a better idea of why this enzyme or this, if we can be really confident that this patient is indeed sick and that we have a confident way to detect it, right? And so if we look at this experiment, we can see that in the patient versus the 2 controls, we have the individual response of GA2 versus ceramide. And note, this is not a quantitative assay, so it's semi-quantitative. But as we use the ratio of this, we can see a huge elevation in that ratiometric response that makes it very diagnostic of something with the patient. And then we can go back and say, most likely this is the area that's happening where you have insufficient activity, probably related to a different isoform of alpha-galactosidase A. And so what we're doing is whole genome sequencing to confirm that. And because there's so many different variations of this gene. And so what we'd hope is that this work would allow us to then have a new test. And we've just recently published this work in November in the Journal of Mass Spectrometry and Advances in the Clinical Lab. So you can read over that work if you'd like to. And with that, I will close thanking for those people who we worked with on this. Specifically, I showed work from students and a postdoc in my lab. Hoda did most of the work on meningioma and Fabry disease, and then Iqbal did a lot of the work on the prostate cancer. Our collaborators, Ranjan Pereira for prostate cancer, Bill Clapp and Jesse Krisak, and Mark Siegel for both Fabry and meningioma. And of course, thank for the funding agencies. And I'd like to thank you all for your attention. And this is just a picture of the campus at the College of Medicine at the University of Florida.
Okay, thank you very much, Dr. Garrett. An amazing presentation. Now I'm going to read the question from YouTube, and we can discuss all about this. And so the first question is, regarding the prostate cancer study, if the data was normalized using total useful signal, what was the function of the internal standard?
Good question. So the function of the internal standard is to help us assess and it's multiple internal standards. So we added 15 internal standards. It's used to assess extraction efficiency across the run. So we calculate the error that might happen from A to B. And that allows us to see if we have any decay happening in a run, but it also allows us to see that no samples are overly concentrated. So if you in urine, you could have a very concentrated urine, and that might cause total ion suppression, and that would affect your internal standard. So you'd see a very big dip in the response of your internal standard. So really the goals of, and the internal standards represent across the entire chromatograph range that we're measuring. And so it's really just a way to validate what's going on.
Right, and how was the data normalized?
The data was normalized to total ion signal, but we measured the internal standard response before normalization.
Okay, perfect. And did you observe time-dependent signal drift during the experiment? If that was the case, what kind of measures did you take to correct it?
In this run, we did not see any internal standard drift. In other studies, when we see that, we have to decide what's going on with it. And so if we see a decay over time, that means the instrument's dirty, and we would need to vet and clean the instrument so that we retain our signal, and we have to rerun our other samples. If we see a signal that might jump up for one sample, that just could mean that the person made an error and added the internal standard twice to that sample. And that could mean you have to throw out that sample. So in this case, we didn't see any change over the course of the run, whether it was, and these are over multiple runs. And so all of them showed the 15% RSD or less.
Right. Dr. Maria Eugenia Monge has raised his hand. Maybe you can allow your microphone.
Yeah, I don't know. Can you hear me? Hi, Male.
Hello.
First, many thanks, Dr. Garrett, for your excellent presentation. Hopefully, we can all see more of these successful translations to the clinical settings of untargeted metabolomic studies. That's the hope for, I guess, from most part of our community working in untargeted metabolomics with health applications. And I actually have a question regarding the first part of your presentation, the genital urinary cancers results. They are all based on pilot studies, like comprising around 10 samples, right? From 10 individuals in each case.
40, typically at 40. We're at either 20 to 40 in most of the runs. And so this example includes at least 40 prostate cancer and 80 controls. We didn't at this time have enough bladder and kidney collected because we just started adding those. So we do now have enough of those that we can add those in. But typically our pilot studies for human are somewhere between 20 and 40 samples.
And so, like many things, and following up on this question, have you thought about like including or taking into account the different ethnicity, diet, lifestyle, and the effect of the exposome on your large scale studies?
Of course, yeah, that's part of what, yes, that's part of what we want to do in this grant proposal we're submitting, is to include a broader ethnicity group. And I don't actually know the ethnicity of these samples because we were blinded on purpose. And so we know the age, and we could probably get the ethnicity, but we don't have enough samples across them to know whether there is a uniqueness between certain types of people. But that's what our new study will have 200 per group.
Right, and they are all from the US.
That's correct. And they're all probably from the state of Florida. And so it could be,
okay.
Yes.
Now, hopefully we will have more of these studies, including like different geographical regions, right? Like to evaluate that. Like, I guess that's important to think in, like, in terms of translating results to clinical settings and making like global diagnostic tests, like that.
That's very important. I agree. And I'd love to, the only way to do that is to have international collaborations where people want to do stuff, because it's really, and that's because, I mean, it's hard for a single lab to run 10,000 samples a month, right? But multiple labs could run 1,000 samples a month, right? And then you could sort of put that data together, which would be really cool. And I'd love to see that as a future of how we do this kind of biomarker discovery. But that means everyone has to get funded, right? And that's the hard part.
Yeah, and also in those terms, like also the community may have to use similar quality control practices, right? Like to be comparable. And
that's very true. Yes.
Thank you very much.
Yeah, thank you.
Right. I have another question regarding the two biomarkers that you don't know the identity, the two unknowns potential biomarkers. How do you detect these features as potential biomarkers?
Gotcha. So these were detected and picked. We use MZMine as a way to do feature selection. And so but we also use a technique called blank feature filtering that filters out noisy peaks from the data based on the signal intensity at that exact time in the chromatogram for that exact signal. So we want to sort of eliminate those potential noise features. So then at this point, it then becomes a massive retention time. And then we go back to say, does it have a chromatographic envelope that's clean? And indeed, does it have an isotopic pattern? And then I can go back and say, okay, if I see what ion is it, right? Because it could be an N plus sodium, or it could be an N plus H, or it could be an N plus ammonia. It could be lots of different ions, which is the frustrating part about mass spec. And so you have to look for evidence for other isotopes or other adducts that might tell you what ion you have. And we did that. And so this one, we think they're two steroid hormones, but we don't know for sure. We have to find the standards and then validate them.
Yes, but I wonder, how did you arrive to these two features instead of others? I mean, did you apply any strategy for feature selection, or the most significant changes among the classes?
Oh, I got your question now. Okay. This was all through statistical analysis, and we used a combination of principal components analysis, partial least squares discriminant analysis, and support vector machine to come down to these targets. And these are only 2 of the 10, and this one represented the 10 that were the most significant. And so we used a combination of different statistical programs to sort of make sure we had the right feature to that we were confident that yes, indeed, it was true. And then this is just a simple way to show that, right? I didn't show you all the stuff that went into that part of it, but yeah, we used multiple statistical programs to make sure that we had something that definitely was true.
Thank you. Soledad has raised her hand. Do you have a question?
Thanks, Malena. Thank you, team, for your outstanding presentation. I was wondering how do you control the effect of medications? A long time ago, I was reading some papers, and you know that those peaks are present in the clinical samples. And how do you deal with this situation?
The good thing typically is that if you have enough controls, or not enough controls, enough biological replicates, typically those features tend to wash out because they're only present in, say, a few patients. And so they typically hopefully they end up being excluded once you do feature selection, because they're only indicative of a specific state. But in cases where we've seen them and they've caused issues, we typically just exclude the sample, because in some cases, the signal might be too intense, and it might cause ion suppression for other peaks, and then it makes that sample unusable. And I know in a clinical world, you may not be able to you can't exclude samples in a clinical world, right? You have to run them. But you can tell a patient that your sample isn't useful for this test for these reasons. And you can have exclusionary criteria to exclude samples to say we need to collect a new sample. And it could be they just said, like I know with if you get your triglycerides measured, right? They ask you if you had a burger before coming in, or if you ate anything, right? And if you ate something, you really have to come back in when you're fasted so that you don't have that. So, but those we typically try to eliminate those, at least in this biomarker study, for that case.
Thank you.
I have another question regarding the lipid match tool. If you want to merge the matrix obtained in negative mode or positive mode, and you have to deal with redundancy because you have different features assigned to the same metabolite, how do you deal with that?
We keep them both. And we just identify one from negative and one from positive. And I'm an advocate for keeping them only because they should. I know in the statistical world, when you have two of the same species, that kind of overweighs your statistics. But I consider them separate because they're run from a separate analysis. And so when you merge them together, if they cluster in the same spot, then I think that validates the fact that that marker is good in both positive and negative ion mode. And I just compiled a data set that has that, where it was this virus study that we had where we definitely saw overlap between them. And I usually just don't get rid of them. I think it's important to keep them myself. And in the end, you might decide to get rid of them in the final picture that goes in your paper. But I'd rather you just identify it came from positive or negative ion mode. And that's for me.
Okay, and if one feature corresponds to more than one metabolites, and because they are co-eluting, for example, at the end, if you want to understand the biological context of the disease, how do you do in that cases?
You have to have better chromatography, much better chromatography. So and the sarcosine study is important because sarcosine is unique from alanine, right? And you'd like to be able to separate them. And because sarcosine does have some other issues for other disorders that are used to diagnostics. So you'd have to, if you really don't know what it is, you have to have better chromatography. And I don't think IM ability would separate sarcosine and alanine because they're much too small. So the only way would be either GCMS or with derivatization, or better LCMS chromatography, Hillick mode or derivatization with chromatography. Yep, it's a question we have to deal with all the time. And you saw I showed both of those as isomers, but that's just one set of isomers. There's lots of isomers. Alloisoleucine is an isomer of course, leucine, and we can't separate it from it. And it's important in certain rare diseases like maple syrup urine disease, where it is the marker for maple syrup urine disease.
Yes, thanks. Maria Eugenia, do you have another question?
Yeah, I have another one. I have a question regarding the tool that you developed for lipid annotation. Do you consider within the tool all the possible adduct that can be formed or generated in the ion source based on the different mobile phases that can be used in a lipidomics study or in a lipidomics method?
Yeah. Yes, indeed we do.
Because
so,
that's
yeah. No, go ahead. No, go ahead. Go finish. Go ahead.
No, because I mean, that was a challenge for us when we had to identify lipids by using an untargeted lipidomics method that we developed in the lab. And we found that it was key to consider all the possible and different adduct ions that can be formed in the source based on the mobile phases that we were using. So I was wondering if the tool was considering all different types of chromatographic methods, because that's can influence and determine the species that you will detect.
Yes, and we do. Yes, you have to. So this one is an example of formate, right? So this would be negative ion M+ formate adduct for PC species, right? Which are often mostly identified in positive ion mode as an M+ H, but it's really a pseudo M+ H because you already have a preformed positive charge. But yes, we look at formate, we would look at ammonia for triglycerides because that's the only thing they form are ammoniated adducts. We would look at M+ sodium, M+ potassium, any kind of adduct that you have. It is definitely written into the database. And if you do change your mobile phase though, say like some people might use like lithiated mobile phases, right? Because often there are some reasons to use an M+ lithium for MSMS. You would have to, of course, change that in the database. But yeah, it's as you said, and it's very important to know which ion it is. And the cool thing is that typically you can use the fragmentation pattern could build up to know that that's what it is, right? Because you often see fragments associated with that kind of loss. But as you also saw, we found some adducts that were unusual that weren't a part of our database. Well, they weren't unusual, but they were the lipids that were attached to them weren't in our database, and that's why we didn't identify them right away. And in cancer, you're going to have a lot of potentially unusual lipids that could show up. So it could be that we're missing stuff still because you get some weird fatty acyl chain attached to it.
Thank you very much. And again, thank you for your great presentation.
And for lipid annotation, can you combine the information obtained in positive and negative mode in order to, for example, know the acyl change in a phosphocholine that in positive mode will be difficult?
You could do that. The software isn't built to do that yet. We would just basically do the identification in negative ion mode and do it in positive ion mode. And so, but that's a good point. You'd like to merge, be able to merge, especially if it's the same retention time and you have MSMS from positive and MSMS from negative. You like to be able to basically consolidate that information into one type of information. The only caveat from that is sometimes in positive ion mode, you have more isomers at a specific retention time than in negative ion mode, just because of you have a lot more lipids detected in positive ion mode than negative ion mode.
Right, thanks.
And the other thing is that this, I don't know how many of you all have used data independent analysis, but it works really well for this case too, but only really for some lipids, not necessarily for everyone. And the good part about it is it's pretty easy to collect because it's just one extra scan, right? And at least on a thermal instrument, it's called all ion fragmentation. The only downside is you don't know what precursor it goes to, but you could get, at least what it does is gives you can give you fragmentation that goes much lower in intensity, so you can get some of the noisier, the peaks with a little bit less intense, but get those fragmentation patterns that potentially match up to it. Although in the end we don't find as many lipids with that way as we do with MSMS.
Thanks. Soledad, I don't know if there's any more question in YouTube.
No, not at the moment. So if there are other questions, we can also forward them to Dr. Garrett later. And thank you, team, for thank you so much, Malena, for moderating this talk also. It was very outstanding presentation. We also thank the audience. They are greeting you and they are also congratulating you for your work. And we are just finishing now our seminar, and we hope you can join us in the last seminar next week. And we will be presenting Dr. Michael Hernandez Mesa for our last seminar of this series of SAEM virtual meetings. Well, thank you, team, again. Thank you, Malena.
Thank you.
Thank you very much.